Okay, you're going to open up your notebooks to the next blank page, and you're going to title your notes Cell Membrane, Structure, and Transport. Okay, so just by the title here, we know we're going to be learning about the structure, aka like what it's made of. And for transport, we're going to be talking about how things move across the cell membrane. Okay, so this lecture is going to be kind of like zooming in and focusing more specifically on just the cell membrane. So let's start first with structure. The cell membrane is made of these little fats called phospholipids, and here is a phospholipid. Notice it's got a little like circular head and these two little tails that come off of it, okay? And when we get a whole bunch of phospholipids together, they're going to form two layers, or what's called a bilayer, okay? Notice up here, we've got a layer of phospholipids and all their heads are facing the same direction and their tails are pointing down. And then down here, all of these heads are facing the same direction, but their tails are pointing up, okay? And when we get a big group of them together, they're going to form like a little bubble. And this is essentially your cell, okay? So these phospholipids are going to form a bubble. And the inside of here, this says water, but the inside of here is going to be cytoplasm. And the outside is going to be the environment, which honestly, cytoplasm and the environment are usually water-based anyway. And so that's how they form. Notice that the heads are always kind of pointing out. So the heads are always interacting with the outside of the cell or the cytoplasm inside of the cell. And the tails are just kind of pointing towards each other. They're just like there in the middle. All right. And so this is a phospholipid bilayer. Bi meaning two, phospholipids, okay? So two layers of phospholipids. And this is going to be what separates our cell from its environment. Now, this picture makes it look like that the only thing present in the cell membrane are these phospholipids, but in fact, that is not actually the case. There's a whole lot of stuff in the cell membrane. These like blue things, there's like huge honking proteins, there's carbohydrates sticking up, there's cholesterol, there's all kinds of different stuff that's actually also in the cell membrane. But if you want to learn more about that, you'll take an advanced science class. Really, we're just going to look at the phospholipids, and we're not going to really worry about the rest of the stuff in the cell membrane. But just FYI, there's other stuff too. So the whole purpose of the cell membrane is it's going to protect the cell from its environment, and it's going to let things in and out. Okay, so kind of like a fence, if you will, right? Protecting a yard from environment, it's going to let some things in and out. Well, like what kind of things? Why do things need to enter and exit the cell in the first place? Like, why is that important? Well, if you remember back to our characteristics of life, one of them that we talked about was maintaining homeostasis, okay? And homeostasis is just regulating the internal conditions to keep them constant, right? Like you can think about our bodies, for an example. Overall, our bodies want to be roughly 98.6. We don't want to get too hot or too cold. So either we sweat or we can like shiver and get goosebumps to kind of return our temperature back to that 98.6 if it gets too high or too low. Same thing with our cells. Our cells are going to keep that internal environment, the stuff inside of the cell membrane, is going to want to keep it constant. Okay, it's going to try to keep it balanced. We don't want too much stuff like building up or not getting in and out. Okay, so it's going to try to keep things constant and balanced. So the way that cells do this is they're going to basically, some things are going to come in and out across our cell membrane. Some things, but not all things. And the term for this is semi-permeable or selectively permeable. Both of these terms are used, and I will probably use them both. They can be used interchangeably. They mean the same thing. Permeable means things can pass through. So if it's selectively permeable, that means it's going to be selective. Some things can, some things can't. Same thing with semi, okay? If we look at this diagram right here, we can see that the little blue and red dots were able to pass through to the right, but the yellow dots can't, and they get bounced back and they stay on the left. Okay, so this is how this membrane is semi-permeable. It's allowing the blue and the red through, but not the yellow through. Okay, the same thing is going to happen with your cell membrane. Some things are going to be able to pass through and other things cannot. So here are some things that might be trying to get in and out of your cell. Things like nutrients, oxygen, carbon dioxide, water, waste, proteins, and some other ions or elements like sodium, potassium, calcium, things that maybe you're used to seeing on like nutritional labels and stuff, right? Okay, so these are things that might need to enter and exit our cell, okay? And so let's look at how these things actually get across the membrane. And in order to understand how things move across the membrane, you have to understand concentrations, okay? So we're gonna pretend that this rectangle right here is like a cell or something. And this little pink line is a membrane, a selectively permeable or semi-permeable membrane. And the little circles are oxygen. So where is there more oxygen? There's more oxygen on the left side, very clear to see. So we would say there's a higher concentration of oxygen on the left side and a lower concentration of oxygen on the right side. Okay, very easy, cool. Well, as it turns out, just on its own, this oxygen is naturally going to go from the area of high concentration to the area of low concentration. So the oxygen is going to move from the left side to the right side across the permeable membrane, okay? This, by definition, is called diffusion, okay? So the diffusion is whenever we have the movement of something from an area of high concentration to low concentration, like our oxygen right here. It diffused across the membrane. Here's another example, but not using oxygen this time. This time, we're using little molecules of dye. This is a semi-permeable membrane. I know it looks like it's got holes in it, but when you think about it, there's just got to be little gaps in between the membrane, the phospholipids for things to bust through. So think about like a really super crowded area full of people. Like, is it hard to get through? Yeah, but can you like find a little way through? Yes, that's like your membrane, okay? It's full of phospholipids, but certain molecules can like kind of bump their way through. They can find little gaps and bump their way through, okay? So here we've got our dye, and here we've got our membrane, and we've got all the dye on the left and none on the right. So we've got high concentration and low concentration, so the dye is gonna diffuse across the membrane. And then here now we've got some on the right, and it's going to keep going until we have equal amounts of dye on both sides. And the fancy pants vocabulary term for that is equilibrium, when we have equal amounts on both sides. And once we get equal amounts, then we're good. There's not going to be really any more movement. Okay, so it's not going to be all of a sudden that there's a whole bunch on the right side anymore. It's going to stop when things equalize. One last picture here. So here we go. We've got the outside of the cell on top and the cytoplasm, the inside of cell on the bottom. Look, here's our phospholipid bilayer. And then we've got just some hexagons representing something that's trying to get through. So where is there the higher concentration of hexagons? Inside of the cell or outside of the cell? It would be outside of the cell, right? Okay. And so which way are they going to move? They're going to move down, right? They're going to move to the inside of the cell. Okay, so here we are. We got a super high concentration. We have a very low zero concentration. And then look at our blue hexagons. They are moving through our membrane. And so now we're getting some on the inside. And now here as more time passes, notice we have equal amounts of hexagons outside the cell and inside the cell. And so this over here on the right-hand side would be equilibrium. So there you have it. Now you know what you need to know about membrane structure and phospholipids and also transport across the membrane.